So welcome everyone. In this presentation, as Aphrodite very kindly already told us, we are going to discuss a very brief overview and the key concepts of individual patient data meta-analysis. So the outline of today's presentation, we are going to see the backgrounds and then make introductions with IPD. We will then learn why we should use IPD and what an IPD dataset looks like. We're going to discuss the strengths and limitation. And then George will walk us through step by step on how we can conduct an IPD meta-analysis, softwares and tools that are available for conducting an IPD meta-analysis, and what reporting standards should be followed. So let's start with a poll. So how familiar are you with IPD meta-analysis? It looks like we have a mix, so no problem at all. We will go through the basics and jump a bit deeper as well. Okay, and another poll. So what do you think? An IPD meta-analysis is only about rerunning the same analysis with more data. Is that true or false? Okay, so I'm glad that so many said false, and because now we will discuss the real power of IPD. So let me start with some background information. So meta-analysis is an established statistical technique that combines all the relevant data that address a specific research question, comparing two interventions. So the data often come in the form of aggregated data, meaning that the results have already been summarized across all participants within each study. These are typically reported as effect estimates, such as the risk ratio, odds ratio, or mean differences, depending on the nature of the outcome, and these come along with their measures of uncertainty. We can also use meta-regression to explore why intervention effects differ between studies and to identify any conditions under which an intervention may work better or worse. So aggregated data meta-analysis, what it does is to combine the summary results from different studies and estimate an overall average treatment effect. So in other words, what we do is we bring together multiple sources of information, but for now, we only use data that have already been aggregated at the study level. But relying only on aggregated data alone might pose restrictions to the kinds of analysis that we can do. So first of all, we are limited to whatever each study chose to report. So important outcomes, for example, or covariates may be missing. Then the within study complexity can be ignored. And with the term within study complexity, we mean within study variability, any interactions or any nonlinear effects that at individual level can be modeled, but when we use aggregated data cannot be modeled directly. And finally, there is a risk of ecological or aggregation bias, which means that relationships that are observed in the aggregated data do not necessarily hold on the individual patient data. So this plot is an example of ecological bias. In this graph, its point is a study. So the x-axis corresponds to study's average covariate, which in our case is the average age in each study. And the y-axis in this graph corresponds to the study's overall treatment effect. In our case, this is measured with SMD. So if we consider to fit a line through these points, then we are relating the treatment effects with SMDs with the average age per study. So in our case, this tells us that studies with older patients show a bigger benefit. But what if we had the raw data per study? And these are denoted now with these purple lines. So in that case, now that we have the whole dataset from each study, then the trend that we observed does not reflect what happens within studies at the individual level. It seems that there is no relationship such the one that we observed earlier. So in this case, a meta-regression suggests, for example, that older patients benefit more, but that conclusion is misleading because the relationship can be entirely due to differences between studies and not due to cannot be due to a true effect of age within individuals. So we have just seen how relying on aggregated data can lead to ecological bias and, of course, misleading conclusions about which patients, in our case, benefit most. But what if instead of just having one summary number per study, we had the raw data for every participant in every trial? What that would allow us is to model the effects directly at individual level, properly account for the variability and any interactions, and of course avoid all these fallacies like aggregation bias. And that's exactly what individual participant or individual patient data gives us. So in the rest of this presentation, we're going to focus on individual participant data or IPD, and we are going to see what is this, why it matters, and how to use it. We are going to walk through a published meta-analysis on as our empirical example, and keep coming back to it as we introduce the key concepts and methods. So the aim of this published meta-analysis was to compare the successful and unsuccessful EVT, that is the intervention and comparator, for reducing systolic blood pressure in patients with acute ischemic stroke. So reducing systolic blood pressure is outcome, and the participants are patients with acute ischemic stroke. Also, AIDS was included as an important covariate in this analysis, and in our case here, we are using a subset of the original dataset only to illustrate how an IPD meta-analysis can be carried out. So this is what aggregated data looks like in our example. Each row is one study, and for its treatment arm, okay, so each row is a study, and for its treatment arm, we have the summary statistics, the mean systolic blood pressure, the standard deviation, and the sample size per arm. So for every study, we only have these three. For every arm and for every study, we have only these three pieces of information and not the raw measurements for each individual. So using this aggregated data, as we call them, we can conduct the meta-analysis using the typical approaches. So in this case, we have combined all this information under both a fixed and a random effect model. And we have used the mean difference as effect size, and we can see that based on our results and the pooled estimates, that the experimental, the intervention is favored in, compared to control. And we can see that heterogeneity is low, and this is probably due to this large confidence interval of the first study. So now let's focus only on study 1 and study 2. And for this, we only know for its arm, as I told you before, only this information, only these three pieces of information per arm. But behind each of the lines, there are hundreds of individual patients. So each patient has its own systolic blood pressure at 24 hours, their own age, and any other characteristics. And all that rich detail gets collapsed into just a mean, a standard deviation, and a sample size. So these three clusters here, denoted with different colors, correspond to the three first studies. So this is the first study, the second study, and the third study. And each of the studies includes its patients. And for each patient, we know to which study they belong, their participant ID, which intervention they received, their individual systolic blood pressure at 24 hours, and their age. So with this structure, we can analyze treatment effects directly at individual level. We can adjust for age or any other covariates. And the aim now is to analyze all these data that we have available. So this is what IPD meta-analysis allows for, to synthesize evidence across studies using the original participant-level study data. So there is a clear increase in the use of IPD meta-analysis. There is one large review that identified 760 IPD meta-analysis published between 1987 and 2015, while a more recent review found 323 IPD meta-analysis across 21 different clinical areas, and these were published between 1991 and 2019. So IPD meta-analysis is being used widely and across many fields. So it is considered as the gold standard approach for evidence. And the reason behind this can be that with IPD, we have the maximum level of information available. So we can see in this graph that the more that we process and compress the data, the less information we have. So the more information we throw away till the synthesis of aggregated data. So what are the strengths of an IPD meta-analysis? First of all, we can deal with ecological bias. So with IPD, we have data on each individual patient and not just the study averages. So this means that we can properly adjust for important patient-level factors like age in our case or any other covariates of interest and avoid in this way ecological bias. Then we can reanalyze all eligible RCTs using the same analysis model and making the same assumptions. So what we do, instead of relying on whatever analysis each trial team happened to report, we can now reanalyze all original RCTs in a consistent way. So we apply the same outcome definition, we use the same covariates, and we use the same statistical model across all studies. And in this way, we make our results more comparable and less dependent on the reported trial methods. Then another advantage is that we can mitigate bias. And what I mean with that is that when we have access to IPD, we can check the data quality, we can verify aspects like randomization, we can explore any protocol deviations, and all these checks can help us reduce any different types of bias that can step into our analysis when we use only the published summaries. Then a basic problem in general in statistics are the missing values. And the imputation techniques, for example, that are available for missing data can only be applied when we have the individual participant data available. So having these kinds of data available, we can use advanced methods like the multiple imputation methods. And in this way, we can handle missing values at the patient level, and we can take into account any relationship between variables, and this gives us a more flexible analysis, and we gain also power. And finally, one of the advantages is that we can handle rare and zero events more appropriate than with summary estimates. So when outcomes are rare or when some treatment arms have zero events, then the standard methods for aggregated data might perform poorly or even fail. For example, double zero studies in some cases are not considered in meta-analysis. But when we have IPD, we can fit more proper models for rare events, for example, hierarchical models, and these models can use the information more efficiently and reduce any bias in the estimated treatment effects. Okay. However, on the other hand, there are some limitations. The first is that conducting an IPD meta-analysis can be time-consuming. So typically, an IPD meta-analysis takes much longer than an aggregated data one. And this is because we have to contact investigators, we have to set up agreements and walk through multiple data sets. And projects like this are collaborative. They require different parts to collaborate in order to conduct an IPD meta-analysis. Then it can be costly and resource intensive, as it requires substantial resources, managing the project, for example, transferring the data, cleaning them, harmonizing variables. All these procedures might take a lot of time and require a lot of expertise. Then there are issues with IPD availability. So sometimes the data are only accessible through sponsors platforms, and this can pose restrictions on how we can analyze them, how we can combine them. For example, if we have to work on sponsors platforms only, we might have limited software options or even no ability to export full datasets. And finally, even with IPD, we might have key covariates that may be missing. So even if we have all the information available for every patient, some important variables simply might not have been collected in some trials, or they cannot be shared, and all these might limit the analysis that we would like to do. So how can IPD be retrieved? We usually start by contacting the original trial investigators and asking them whether they're willing or able to share their datasets. We may also approach sponsors or pharmaceutical companies who often hold the trial data, but there are also currently the data is available through data sharing platforms or repositories. For example, for certain diseases or certain outcomes, we might be able to request access for a specific project. And there are also registries and networks, there in some cases, where we can update data that already have been used in multiple trials. Okay, so once we obtain our IPD, or even if we have both IPD and aggregated data available, how can we synthesize all this together? So I'm now going to hand over to my colleague George Seitidis, who will explain more about it.
Thank you, Sofia, for the introduction. Now we are going to see how we can synthesize the IPDs. There are two principal approaches for conducting an IPD meta-analysis: the two-stage approach and the one-stage approach. In the two-stage approach, each study is analyzed separately, and the study level estimates are then combined using standard meta-analytic techniques. In contrast, the one-stage approach analyzes all available studies within a single framework, and at the same time, it accounts for the fact that participants are randomized within studies. This framework can model complex relationships, adjust for patient-level covariates, and investigate treatment covariate interactions. It is important to emphasize that under the same assumption, these approaches are generally expected to give similar results. The main distinction lies in how the data are handled and how flexible the model can be. Let's focus in more detail on the two-stage approach, which is the method that it is commonly used in practice for conducting an IPD meta-analysis. This approach closely mimics a conventional aggregate data meta-analysis, and it can be described in two main steps. In the first step, we calculate the effect size for each study separately. So, for every study, we analyze the individual participant data to estimate the treatment effect, such as a mean difference, a odds ratio, or hazard ratio. To obtain these estimates, we typically use a generalized linear regression model, for example, a linear regression if the outcome is continuous, a logistic regression if the outcome is binary, etc. At this stage, an important advantage of IPD is that these effect estimates can be adjusted for several covariates, such as baseline characteristics, prognostic factors, and of course, using a consistent modeling strategy across all studies. After we have an effect estimate and its standard error from each study, we then pull these effect estimates using the standard meta-analytic techniques. The key difference is that our study level estimates now come from a harmonized analysis of the raw data in which we use the same set of covariates across all studies. This typically improves the robustness of the findings and mitigate potential biases. So let's describe the two-stage approach graphically. As you can see, we have 6 studies, so we have 6 IPDs. At each study, we fit a linear regression model, since we have a continuous outcome, the systolic blood pressure, in order to estimate the mean difference and the standard deviation from each study. After estimating the 6 mean differences and their standard deviations, we combine these estimates using the standard meta-analytic models. We can mathematically describe the two-stage approach in our umbilical example with this model equation. By applying this model equation to our data, we will get the following results. We now see that the confidence intervals of the pool estimate are slightly reduced, so we gain precision by using IPD. We also see that we have a small level of heterogeneity. This analysis is based on unadjusted estimates. If we want to adjust the systolic blood pressure for prognostic factors or baseline characteristics, this can be easily done by simply including these characteristics as additional covariates in the regression model. In this example, we have adjusted the systolic blood pressure for the patient age. After adjusting for age, we observe that the direction of the treatment effect remains the same. The intervention still appears beneficial. However, the magnitude of the effect is now reduced since the pool mean difference is smaller compared with the unadjusted analysis. And of course, the confidence intervals have changed slightly. This change is partially explained by the behavior of the heterogeneity parameter. In the adjusted analysis, tau is estimated to be 1.55, indicating an increase in heterogeneity compared with the unadjusted model. In other words, once we account for age, we see a somewhat smaller overall treatment effect and more variability in the effects across studies. This illustrates how adjusting for prognostic factors can influence both the pooled estimate and the degree of heterogeneity in an IPD meta-analysis. Let us now move to the one-stage model, which analyzes all studies simultaneously within a single framework. This is typically implemented using generalized linear models, generalized linear mixed models, or hierarchical models. A key requirement is that the model must respect the data structure. Different patients come from different studies, and this randomization must be accounted for in the model specification. Within this framework, we can easily adjust for prognostic factors, just as we did in the two-stage approach, and also we can incorporate effect modifiers and explore interactions between treatment and covariates. In general, the one-stage approach is highly flexible, and this allows us to handle a wider range of situations. For example, we can more easily model other types of outcomes, such as longitudinal data, longitudinal outcomes, where each patient has multiple repeated measurements, or we can deal more effectively with rare events by using more sophisticated models. However, this flexibility comes at the cost of greater model complexity. These models can be more challenging to specify, more challenging to fit, and they often require more statistical expertise and computational resources. Let's see the one-stage approach graphically. Now, the IPDs are simultaneously analyzed, and we assume that the mean difference of the studies share a common distribution. Usually, this distribution is the normal distribution. The center of this distribution is the meta-analyzed pooled effect that we want to estimate, and the variance of this distribution is the heterogeneity. One-stage modeling, in our case, requires fitting the following model, which may look the same with the previous one, but it is not. The previous model requires fitting regressions to all studies separately, while this one is applied to all studies simultaneously. A key consideration here is how we handle the intercept. Intercept is vital in this approach since it is the parameter that randomizes the patients based on the study that they belong. There are three options here. The first option is to assume a single intercept for all studies. So instead of alpha I, we have a constant alpha. By assuming common intercept, we assume that all studies have the same baseline value on average. So in our example, this means that all 6 studies have the same systolic blood pressure levels on average at baseline. This is a quite strong assumption, and it is not recommended for most of the cases and rarely used in practice. The second option is to assume a study-specific intercept. By doing this, we assume that each study has its own baseline, so each study has separate baseline systolic blood pressure levels. Now, we assume different values for each study without making any assumption about the distribution of the baselines across studies. Because of this property, this approach could be considered as a standard approach. The third option is to assume a random intercept. So in this case, we assume different baselines across studies, but now these baselines come from a common distribution. Again, this distribution is a normal distribution usually. This approach is useful when you have studies with few data, for example, studies with less than 30 patients. With the one-stage model, we can easily adjust the estimates for prognostic factors by including them in the model equation. We can assume that a predictor has the same impact across studies, or we can also assume a study-specific or a subgroup-specific effect, so that each predictor has different impact at each study or subgroup. Furthermore, we can also add interactions between covariates and treatment groups in order to adjust for potential effect modifiers. What we have done is that we have applied the random effect approach under different settings, and we compare the results from the one stage with those from the two-stage approach. What we can see from this table is that in all analyses, the confidence intervals of the IPD meta-analysis are smaller than the confidence interval of that of the aggregate data meta-analysis. So by using the IPDs, we gain precision. What we also observe is that the results from the two-stage analysis are similar to those with the one-stage analysis. For example, by looking at the second and the third row, we see identical confidence intervals and a quite small change in the effect size. This is because we had made the same assumption for these models. We conclude the same if we adjust for the patient age. Keep in mind that these approaches give in general the same results under the same assumptions, but assumptions have to be the same. Even a small change may differentiate the results. As we did in the last case, we do the same analysis, adjust for age, and also use the same intercept, but we used a different estimator for heterogeneity. We used, for example, maximum likelihood instead of restricted maximum likelihood. By doing this small change, we see that the results differ. To summarize the discussion so far, I will briefly compare the one-stage and the two-stage approaches. The one-stage approach analyzes all studies simultaneously within a single framework. It offers flexibility, but at the cost of increased model complexity. This is typically done using a generalized linear model, and this model can be computationally intensive, but they are particularly useful when we want to explore interactions or potential effect modifiers or model complex structures. On the other hand, the two-stage approach is a two-step process, is currently the most commonly used method in IPD meta-analysis. It uses standard meta-analytic techniques, and it is simple to implement and also to interpret. As we saw in the example, it will generally provide the same results with the one-stage approach under the same assumptions. In practice, this means that the choice between one stage and two stage is often driven less by which is the better approach and more by what is needed for the analysis. So the two-stage approach can be used as a pragmatic default option, and the one-stage approach when additional flexibility or more complex modeling is required. People often ask a very practical question: which approach should I use in my analysis? This table summarizes some guidance. In situations where all studies provide IPDs, both approaches can be used, and it is expected to give similar results under the same assumptions. So the choice can be based on familiarity, software availability, and of course, personal preference. When we need to combine IPD with aggregate data from studies that did not provide IPD, the two-stage framework is usually preferable since it is more straightforward on how you should combine these estimates. One-stage approach can be also applied, but it is considerably more challenging and requires some expertise. When you collect the data, you may encounter the case where some IPDs are available in a Sposon platform and some other IPDs are available to your computer. In that case, it may be impossible to merge all the datasets into a single file because the platform does not allow to export patient-level data. In this case, you can only apply the two-stage model. The same holds if IPD are available in Sposon platforms and you also have aggregate data. Regarding software availability, the two-stage approach in the software R can be used using the standard meta-analytic R packages such as Meta or Metaphor, and also there is a plethora of R packages for the one-stage model. It should be noted that there are also packages in other platforms such as Stata or SAS. Regarding the reporting of IPD, there is a specific reporting checklist for systematic reviews and IPD meta-analysis, which is the Prisma IPD state. It includes the main 27 Prisma items plus 3 extra ones for IPDs. These ask to describe how we found the data, how we requested, how we collected the IPD, and also we should describe how we checked, how we verified the data, and which IPD we actually had, which IPD were available in our analysis, and what type of analysis was conducted. Did we use the one-stage approach, the two-stage approach, what assumptions we made, and why? To conclude, I would like to discuss some practical suggestions. Firstly, when using IPD, please do not dichotomize continuous variables since we lose power. Secondly, centering usually improves estimations. If you are not familiar with IPD, maybe it is better to use the two-stage approach. It is safer and it is less likely to do methodological mistakes. Of course, in the presence of rare events, one-stage approach is preferable since you can apply more sophisticated models. Lastly, before conducting an IPD meta-analysis, consider the added value of the IPD meta-analysis and if you expect the IPDs to give different results. Since if the research question can be already answered, maybe it is not a good idea to conduct an IPD meta-analysis since it requires a lot of resources and it is a time-consuming procedure. A power calculation may also help you on planning an IPD meta-analysis and see if you expect to have enough power to reach a meaningful clinical decision. This is some references and useful resources that we used in our presentation. Thank you very much for your attention. We are happy to answer any question.
Thank you both. This was very interesting. So we have a couple of questions in the chat. We have two questions about the two-stage unadjusted IPD. So Katie asks, I'm not sure why the confidence interval gets more precise with the two-stage unadjusted IPD. Shouldn't this essentially be replicating a meta-analysis with aggregated data? And Wolfgang asks kind of the same things. He says that the mean differences we use for the aggregated data meta-analysis are exactly the same as the ones we get from fitting the regression model you described. So the results should be identical.
We gain precision in this example by using IPD because we estimate better the uncertainty. So we have smaller standard errors and we estimate better the heterogeneity, and this is the reason that we gain slightly reduced confidence intervals. So it is not guaranteed that always we will get exactly the same results.
Okay, great.
Yes, and may I add that, okay, we gain precision because, as George very well explains, that's the published aggregated data might often ignore some of the data of the information that we have available, right, George, in with IPD. So these are better models, and might be why we have better estimates, and then we gain precision.
A better statistical analysis might be within its trial. Yeah. Okay, thank you. So Young asks, can I merge IPD data with aggregate data? If some studies have IPD data available, but others don't, let's say we could use two-stage approach for a two-stage approach for this setting, which is, I think, it's quite common issue.
You can use both methods, both one stage or two stage approach. The two stage approach is more straightforward on how you can combine these estimates. So you analyze the IPD separately, you get the estimates, you have the estimates for the aggregate data, and then you just use the simple meta-analysis model. In the one-stage approach is a more complex procedure, so it requires some expertise in order to do it. It is feasible, but sometimes it's challenging.
Okay, so Giovanni asks, why do you need to adjust for age in the two-step meta-analysis when this confounder usually handled in the randomization process within each trial?
Yes, I can
I-
take that. Yeah. So, okay, there are, well, the randomization handles confounding in a trial on average, but an individual trial, you can still have chance of any imbalances between groups. So you can adjust. You're not fixing any bias, but you're trying to reduce any noise. So using all these adjustments, you make the results more comparable across trials.
And
Yes, Georgios.
also, uh, studies are different, so you don't have the same mean age across studies. And also, if you conduct a meta-regression, you may have ecological bias, as Sofia presented at the beginning. So by using the IPDs and adjusting also for age, you don't have these type of biases. You account for the fact that studies has different characteristics, and you also avoid biases.
Okay. What does the power calculation involve exactly? By mean he.
Okay, so power calculation for an IPD meta-analysis is a way of checking in advance whether if collecting and combining the raw data from the multiple studies that you might have will probably give a clear answer to the research question. So using the information that is already available in the reported trials, you can estimate, there are some specific statistical methods that are published, especially this is straightforward for continuous, I think, outcomes, and more difficult for dichotomous and time to events outcomes, but there are methods published. And you try to estimate the chance, let's say, that an IPD meta-analysis will successfully detect a true effect or difference. So the power analysis will tell you if it is worth proceeding with an IPD meta-analysis or not.
Okay. Young asks, could we check interactions of variables from two-stage method?
Uh, it is more difficult to do it. So
Yeah.
it is like regression. So you have the same limitations, more or less.
Do you need ethical approval for an IPD meta-analysis?
Well, I'm not sure, but I think that's not, because these are trials that have already been conducted. So you might need some, you might have issues with obtaining the IPDs, and uh, I'm not sure how to answer this. There might be some problems with accessing the IPDs, but I get if these are anonymized and if there are procedures followed, and I think that there are protocols for that. I think that this can be resolved. But I don't consider this as ethical approval as, for example, we consider in trials, in clinical trials. I don't think that's something like
I
this.
guess you might need some permissions.
Yes.
You might need to be given some permissions to access the data.
And
Um,
and also some data, sorry for interrupting, some data, if the IPDs are secured also in some servers. Some known servers such as Vivli, you can upload the IPDs and these data are secured and you cannot download them you can only access the data through the platform.
And I see that Philomena answered that no ethical approval is required, and approval is required from the study investigators, yes, to give the data. So we agree.
Could you explain a little more about the hierarchical model for error outcomes?
Hierarchical modeling, it is usually in the Bayesian framework. So if you want to be more flexible, usually you go with the Bayesian framework and you can incorporate also past knowledge. And usually we use penalized methods. so such as first regression, and so you can do it with more flexibility in the Bayesian framework. So this is a hierarchical model for meta-analysis with rare events.
Yeah, so it is better to apply these models when you have the full data set and the IPD, and you do any adjustments in there, you obtain the treatment effects more properly, and then you can summarize all this in the meta-analysis.
Okay, a clarification from Wolfgang, I guess, from the two-stage meta-analysis. He says that I guess we expect no difference between the two-stage analysis and the classical meta-analysis because the treatment effect estimate, which is in these cases, difference, is exactly the same with the one we get from the regression model, and the standard error is also identical. So I guess we expect no differences. So yeah, I don't know if there are any comments on this one.
We expect to be identical, but it is not always the case. So you may have some computational issues, or so it is not guaranteed that always have the same results.
Okay. Rode says that trials can be small and therefore have some imbalance in covariates. What about adjusting for baseline, in this case, systolic blood pressure, as we know from AMCOVA, that can improve precision?
Of course, you can adjust for a lot of different covariates. Here it is just a illustration, so we use the subset of the data and not the original analysis that we used in the publication. So this is the reason that we only use the NH. We didn't want to make it too much complicated. Of course, in ARCOVA may improve precision.
Okay, so Giovanni asks about graphical representation. So can we have a forest plot when we use a one-step model?
You can, but you have to do it manually because usually softwares are not produce the standard forest plot. But of course, you can do it.
Okay. Yeah, there are some comments about the ethical approval. It says that it's not needed. Okay. Ruth says that some institutions, yeah, the same, do not require ethical approval for IPD meta-analysis. And Rod says that surely a lost opportunity not to use baseline data for the outcome variable analysis. I guess that comes from his previous comment, yes, about the imbalances in covariates. Okay, so yeah, I have a question too. I think one of the main problems we might have is that some trials might not provide the individual data, or we might not find the individual data. So in this case, we can use, I guess, the summary data, the published summary data. So do we expect any differences in if we have the individual data but we don't, and then use the summary data? Do we expect differences because we don't have the individual level data and we use the summary?
If we are using, sorry, George, please go on.
If
And the
the
summary
analysis,
estimates, I guess, if
yeah,
they're
if the analysis has been conducted in a proper way, usually you don't expect to have that much differences. So you may have differences where it is unclear how they handle, for example, missing values, what type of analysis they used. So in cases that it is not clear if the quality of the study is high, so in these cases you may find differences. But if the analysis is properly conducted, usually you don't expect to have that much differences. So that's the reason also if you are going to conduct an IPT meta-analysis, it's not guaranteed that you will get different results.
Okay, thank you. And one last question from Giovanni. Is the risk of bias procedure the same as in aggregate meta-analysis?
Well, it's availability bias. Okay, well, it depends. You can go through. You can use the bias tools. I think that's are already available, but I think that's it depends on which model which model, what type of data that are available, and which of the model you can
I guess
use. I'm not very
yeah.
sure about this.
I guess the risk of bias tool doesn't change. You don't have
No.
to assess any other domain.
No.
Okay.
You don't have
I
any
think
other domains.
it's the same. And you can also verify the results from the publication using the
Yeah.
IPTs.
Yeah.
So this is and you typically report what is the published results and what is the result using the IPTs, and you compare them in order to verify if the analysis is properly conducted and why you observe differences.
Okay. Yeah, Rod says that, sorry if he has not been clear, but his point is emphasizing on the importance of using outcome variable at baseline for adjustment rather than baseline covariates, such as age, as you demonstrated.